So hello everybody, my name is Eli Sprecher. I'm a member of the ESDR board, and together with my co-chair, Professor Bernhard Homey from Düsseldorf, we will try to entertain you during the next half an hour or so. So it's really great to have you with us for another cooking session of our ESDR kitchen. For those of you who are not too familiar with the concept, so this series of webinars takes place every other Wednesday. We have 4 items on our menu. The recipe book, where new techniques are being presented in a simple way. The sweet and sour session, where 2 researchers debate on a hopefully controversial topic in investigative dermatology. Then we have the freshly baked session, where very new data are being presented. And then our molecular cuisine track, where, like today, a prominent researcher in the field discusses her or his professional path. I'd like to mention that this episode is supported by a generous grant from Amgen. So it is today for me a great and a very special pleasure to introduce our speaker, Professor Kathleen Green, who is the Joseph Maybury Professor of Pathology and Toxicology and Professor of Dermatology at the Feinberg School of Medicine. Professor Green serves as the director for Northwestern PhD training cluster in cancer biology and the National Cancer Institute funded carcinogenesis training program. She is a fellow of the American Association for the Advancement of Science, and she is also a recipient of Northwestern University's Martin and Gertrude Walder Award for Research Excellence. She also currently serves as the secretary of the American Society for Cell Biology. She is deputy editor-in-chief of the Journal of Cell Science, and she previously served, as many of you know, as a president of the SID. Her research program is focused on understanding the molecular basis of how cells stick together, not only to provide mechanical strength to tissues, but also to regulate chemical signals, which are important for development and differentiation. Professor Green has a particular interest in tissues, such as the skin, the heart, which are major targets for adhesion-related diseases. At the end of Professor Green's talk, we will have a Q&A session. Please send your question to the chat or the Q&A function. And now, Kathy, it is really a great honor for us to have you with us today. Thank you very much for sharing with us a bit of what you have been cooking along all those years on the role of and importance of epidermal cadherins in human biology. Kathy.
Thank you very much, Eli. It's fantastic to be here this morning to share this edition of the ESDR Kitchen Molecular Cuisine. So when Eli invited me to give this edition of Molecular Cuisine, I decided I needed to inform myself. So I went to Wikipedia, of course, to look to see what the definition of molecular gastronomy is. And here it is. It's the investigation of the physical and chemical transformations of ingredients that occur in cooking. And it has 3 important elements: social, artistic, and technical. And I thought, wow, these are exactly the elements that are important for us in science as well. And I realized that whether I'm at the bench in the kitchen or at the bench in the laboratory, that keeping these elements in balance would be important for my career. And in particular, the mentors and collaborators that we all have really propel us in our career, and the curiosity and creativity that got us into science in the first place and is so important in keeping our passion alive, even as we face challenges such as this time with COVID, which are inevitable in our careers. And of course, I think it goes without saying that technical advances are important, both in gastronomy and in science. But there were some other elements that were important in my career, and I venture to guess in yours as well. Opportunities, some of which are serendipitous, and also choices that we make in our careers are absolutely critical. Now, the evolution of my career from a single-celled organism driven by curiosity to participate in science fairs in the 1960s, all the way to the multicellular organism, that is the Green Lab and all of our collaborators now. Well, that's the question. And actually, this evolution, in my mind, is parallel to the evolution of the questions that we're interested in. And those go to the fundamental question of how did we as terrestrial vertebrates achieve multicellularity through the evolution of cell adhesion. And cadherins are the hero of the story today, and they were absolutely essential for this process of acquiring multicellularity. They first appeared in single-celled organisms in choanoflagellates, and they were organized around the feeding organ, so it was speculated they might be involved in capturing prey. And then in sponges, these cadherins were co-opted by these multicellular wannabes, and they were assembled into primitive adherens junctions, which anchored the actin cytoskeleton to the membrane. Then when we move to vertebrates, we see the appearance of new intracellular junctions called desmosomes, and that's why I'm here today. And with those desmosomes were new cadherins called desmoglians and desmocollins, which are familiar to all of us. And then in terrestrial vertebrates, you get an increase in complexity. As we move out into land, of course, we need new barriers, including the epidermis, which is absolutely essential for our life on land. And with that, we see the expansion of desmosomal cadherins. We have more of them. And also an expansion of the intermediate filament proteins, the keratins that are attached to these cadherins. And so the question that arises through considerations of evolution, and the question we're interested in, is how do you go from simple epithelia to stratified epithelium, which regenerates every 28 days through a process we're very familiar with of proliferation of the basal cells, cell fate commitment to differentiate, transit into the superficial layers, and eventually form the life-preserving and life-essential barrier with new lipids, new proteins, and tight junctions. And what are the roles of the cadherins in this process? Well, to really understand why we got interested in this question in the first place, we need to go back to my PhD days in Washington University, where I was mentored by this gentleman right here, David Kirk, he recently passed away, sadly, who was an expert and a pioneer in using the organism Volvox as a model system to understand fundamental questions in developmental biology. Now, this is a simple eukaryotic organism that has two cell types, and that was really interesting to people interested in cell fate decisions and how these cell types originate. But I was fascinated by something else. I was fascinated by a morphogenetic process, sort of akin to inside-out gastrulation, that Volvox undergoes, and it's called inversion. And I'm going to play this movie for you now, which is a movie of the inversion process that's occurring in an embryo that's still encased within this large mother Volvox. And the reason it has to turn inside out is because the flagella, the basal bodies from which the flagella emanate, are actually pointed into the inside of a hollow ball of cells that is the product of the cleavage divisions. And so in order to be able to swim, the organism has to get those flagella to the outside, and it does so through this process of inversion. Now, why am I telling you, a group of cutaneous biologists, this story? Well, it really was fundamental in my interest in cell connections, because it turns out if you look over here in these scanning electron micrographs, you can see these individual cells are attached at the tips of these long processes. And these attachments actually move, or the cells move relative to the attachments in a sequential fashion to drive the inversion process. And this also requires a cortical cytoskeleton. So you need connections and you need the cytoskeleton, and I was hooked. This was what I was interested in doing. But where to next in my career? Well, I decided to go to summer camp for scientists. So this is the MBL, the Marine Biology Laboratories. And this is where I met my next mentor and had the next opportunity to take a step in my career. And that mentor was Bob Goldman, who was a pioneer in the area of intermediate filaments, which at that time were not very much was known about them. They were the least understood of the three major cytoskeletal elements in non-muscle cells, so actin, microtubules, and intermediate filaments. And I also met this guy, Rex Chisholm, who was an expert in DNA sequencing at the time. That's when we were doing Sanger sequencing with really huge sequencing gels. And he was also an expert in cloning, lambda GT11 cloning. And so I became fascinated with these intermediate filaments and took the opportunity to move to Chicago to do a postdoctoral fellowship with Bob Goldman. Through the years in Bob's lab, I became interested not just in the filaments, but their attachments to these really elegant structures. And this is a desmosome. And you can see that it's almost paracrystalline and in its highly compact and organized structure. And how did these intermediate filaments attach to this structure, and what are they doing? Well, actually, if we go back even farther to the 1970s, when I was an undergraduate student at Pomona College, we could see there was a seed of interest in desmosomes even back then. What you're seeing here is a drawing in the center, which was the result of an assignment I had in my cell biology class to make a molecular model of desmosomes. And at that time, we had no idea what any of the components were. And this model looks very similar to the Singer and Nicholson fluid mosaic model, which at that time had just come out and was all the rage. And you can see these little potato-like structures here, which I call the substance of unknown chemical nature, which I predicted would be linking intermediate filaments to the plaque. And indeed, later on, I became interested when I was in Bob's lab. I did a little sabbatical with Rex and I learned how to clone, and I identified clones encoding this protein desmoplakin, which was in fact the substance of unknown chemical nature. And I began collaborations with these folks here, David Perry from Massey University, who through just the linear sequence of amino acids predicted the structure of desmoplakin, which was remarkably accurate based on the later X-ray crystallographic determination of the high resolution structure of the first intermediate filament binding protein. So this was the intermediate filament binding the main, which Bill Weiss, we did in collaboration with Bill Weiss. We also collaborated with John Stanley, and a group of us compared the structural analysis, did comparative structural analysis of desmoplakin and two other intermediate filament binding proteins, bullous pemphigoid antigen, BP230, and plectin, which had recently been sequenced by Gerhard Wicke. And it turns out these belong to a new gene family involved in organization of intermediate filaments, which was later named the plakin family by Fiona Watt. So right around this time, or a little bit before, I had a big decision to make, another turning point in my career. I was still not independent. I became independent right around the time I was cloning desmoplakin. And we had a two-body problem, not a problem, really an opportunity. Rex Chisholm, the sequencing expert, and I got married. And so we now had two careers and decided, instead of moving away from Chicago and uprooting Rex's 3-year research program, he had just become independent as well. I took an opportunity to take a job in the pathology department, largely because of the enthusiasm of this man, Dante Scarpelli, my first department chair. He decided to take a chance on me. There were very few PhDs in the department, if any at that time, and I decided to take a chance on him. And then later on, I developed really strong ties with the dermatology department and got an adjunct appointment. And this is the current or just recent group that I have strong ties with. And with all of these connections to clinical departments, it helped me start seeing my research through the lens of human disease. And this is something that would not have happened had I gone to a cell biology department. And through the years, the number of mutations that grew in the molecule desmoplakin, as well as other desmosomal molecules, really inspired us to understand what the underlying molecular basis of desmosome-related diseases are. And through these colleagues, who turned out to be collaborators and also now my friends listed down here and many more, we have spent years of really inspirational research into the role of desmosome molecules, the mutations in human disease. So early in my career, I also was inspired by people studying this autoimmune disease, pemphigus. And it was discussed that probably the intracellular junction molecules in desmosomes, the adhesion molecules, were likely the targets in pemphigus. And there was a lot of research around this at the time. And I decided to go ahead and clone one of these adhesion molecules, human desmogliin, which at the time we did not realize would be just one of several desmogliin genes. But over the years, with work in labs like John Stanley's, Masa Amagai was cloning the pemphigus vulgaris antigen at the time, which turned out to be desmogliin 3, and others, we realized that, whoops, there are more than one desmoglian and desmocolin. And this group of us decided to come together and straighten out the nomenclature of the desmosomal cadherins. And now we know that there are 7 desmosomal cadherin genes giving rise to at least 10 isoforms, all of which are expressed in the epidermis. And they're expressed as shown here in patterns. So they're polarized. Some are expressed in more highly in the proliferating basal layers. Others come on as cells are differentiating and become more highly concentrated in the superficial layers here where the tight junctions form. And our hypothesis of why there were so many of these cadherins was that overlaying these polarized or patterned desmosomal cadherins onto classical cadherins allowed tissues to develop new structural and functional complexity during evolution. So this became our guiding hypothesis. And to dig more deeply, we decided to look at, in particular at one of these cadherins, desmoglian 1, which is the one that I cloned years ago. And desmoglian 1 is only found in terrestrial vertebrates. So it's the newest, one of the newest cadherins. We were also interested in it because it comes on as cells begin to differentiate and transit into the superficial layers to undergo this program of differentiation. We wondered whether it was actually involved in promoting this process. And so over the years now, we studied the role of desmoglians in adhesion, along with these folks, Masa Amagai, Andrew Kowalczyk, and a number of others. And then in the mid-years, we became interested in differentiation and the role of desmoglian in this process. And more recently, with Ellie Sprecher and a number of other people, we've become very interested in its role in paracrine signaling. So now I'm going to turn to just telling you a few of our experimental, our forays into understanding the roles of desmoglian in morphogenesis and paracrine signaling. So a lot of you are familiar with this model system, which is a really great complement to mouse models in understanding cutaneous biology. So this is a human reconstructed epidermis made from cells that are put into culture, allowed to get to confluency, lifted to an air medium interface, and then this results in the differentiation into this beautiful structure that's remarkably like human epidermis. Now, this model is highly manipulable, so you can introduce viruses that contain shRNAs to knock down your favorite protein, or you could express mutant proteins. And a number of years ago now, we knocked down desmoglian 1 using this process to demonstrate that desmoglian is important not only for the structural features of human epidermis, but also the biochemical program of differentiation. And you can see here that keratin 10, which is a superficial intermediate filament protein, is no longer expressed appropriately when desmoglian is deficient. And we went on to identify the mechanism by which desmoglian promotes differentiation. It does so through binding to a newly recognized binding partner, ERB2 interacting protein, which in turn interferes with RAS and RAF coupling to attenuate MAP kinase signaling and thereby promoting differentiation. And we also demonstrated that this was not dependent on the extracellular adhesive domain of desmoglian 1. We also became interested in another feature of these cultures, and that is that the cell shapes and the cell sizes were very variable. I'm having some technical issues here. Were variable. And Oksana and Josh were interested in the possibility that these cells were having difficulty moving or transiting into the epidermal layers and wondered whether desmoglian 1 might be promoting or shepherding these cells into the superficial layers. Oksana went so far as to suggest, well, maybe if we put desmoglian 1 into simple epithelial cells, we can cause them to stratify. Now, this was something that I didn't think would ever work, but in fact, she went ahead and did the experiment, and here are the results. So what we're looking at in these videos is we're looking down onto a monolayer of simple epithelial cells expressing either GFP or a fusion protein of desmoglian 1 GFP, and we're looking at the processes and the dynamics of these cultures. And I think if you watch this movie, you can see that there are cells popping up out of the monolayer. They're extruding from the monolayer, then spreading over the surface of the monolayer. And if you look down here, you can see that they're forming these little stratified areas that are expressing desmoglein 1. So in fact, not only is desmoglein 1 essential for this process of delamination, we showed that in keratinocytes, but if you put it into simple epithelial cells that normally never express it, it will cause them to delaminate and stratify. So working with the endogenous machinery, desmoglein 1 is sufficient for this process. So taking it to the 3D, along with work from Sarah Vikstrom's lab and Corinne Neeson's lab, we propose that desmoglein 1, as it is expressed, it works with adherens junctions and actin remodeling to decrease cortical tension in these cells, and that helps push them up into the upper layer, allows them to delaminate and move into the superficial layers. But remember, I told you that desmoglein 1 is actually concentrated up here in the layer where tight junctions are, in the stratum granulosum 2 layer. So we were wondering if it has another role up in late differentiation in this tight junction layer. Now in simple epithelia, tight junctions are in a region of high apical tension. So Josh Broussard and Jen Coetzee in their lab have been using human 3D cultures, and now we're moving into a mouse model of desmoglein deficiency. And Josh has found out that there actually is high tension in the superficial layer and a tension gradient, stiffness gradient from the apical to the basal region in the epidermis. He also found out that like early in differentiation, where desmoglein 1 attenuates EGF receptor and MAP kinase signaling, later in differentiation, it actually promotes ERB2 activity in order to assemble and maintain the tight junction layer. And these all work together, this tension gradient and ERB2, in order to promote the formation and maintenance of tight junctions. So now we've talked about adhesion and we've talked about differentiation. In the last few minutes, I'd like to talk about paracrine signaling. We've done a number of experiments now that indicates to us that the loss of desmoglein 1 can stimulate the production of cytokines and paracrine signaling within the epidermal microenvironment. And we've been interested in both acute loss and chronic loss. But first, I just want to tell you about Jody Johnson's work, where she demonstrated that acute loss of desmoglein 1, which can be stimulated by stress such as ultraviolet, ROS, reactive oxygen species, the peptidoglycans on the surface of bacteria, all these things cause the rapid loss of desmoglein 1. And Jody showed that ultraviolet radiation is one of those, and that the loss of desmoglein 1 stimulates cytokines, and was very interested in whether or not that might actually signal to melanocytes. And indeed, she demonstrated that the loss of desmoglein 1 increases pigment production in the so-called tanning response. And this was published, don't have time to show you all the data, but published this year in Pigment Cell and Melanoma Research. But we're also interested in chronic loss. And this is where Ellie comes in. So Ellie got a group of us together, including me and John McGrath and this whole team of outstanding investigators, to analyze and characterize a new syndrome: severe dermatitis, multiple allergies, and metabolic wasting, or SAM syndrome. Now, if you take cells from patients with this disorder, put them into culture, you can see that they begin to express a Th2 type response, a signature, an increase in cytokines indicative of a Th2 response, which went along with the idea that they had allergies. So we became really interested again in the role of cytokines in the microenvironment when desmoglein 1 is deficient. So in order to study this in more detail and mechanistically in detail, we made a desmoglein 1 knockout animal model using CRISPR-Cas gene editing to remove all 3 of the mouse desmoglein 1 genes. Turns out humans have 1, mice have 3. These animals, the knockout animals, had extremely severe peeling. It looked like pemphigus-like peeling and barrier defect. And they also had a differentiation defect, which was consistent with what we had shown in the human model system. So we use these animals to do RNA-seq analysis in embryonic epidermis, E18.5 epidermis, in collaboration with Xiaomin Bao. Quinn and Hope in my lab did the RNA-seq analysis. And what we found was a very interesting inflammatory signature. Now remember, these are embryos before there's any exposure to the external environment. We saw signatures indicative of chemotaxis of inflammatory cells, secreted antimicrobial factors, and T cell responses. So we wanted to look at this in more detail and ask whether this desmoglein deficient gene signature resembles that of chronic inflammatory or allergic conditions. So along with Johan Gudensson and Alex Tsoi at Michigan, and also Katerina Massaro is involved in this project, we compared the desmoglein knockout signatures to psoriasis and atopic dermatitis. And you can see that there was a higher degree of similarity with the psoriasis patients shown in purple along here than with the atopic dermatitis patients shown in orange. We could also see with further analysis that there was a striking IL-17 skewing or signaling pathway signature, as well as an upregulation of MAP kinase and ERB signaling pathways. So this was also consistent with a psoriatic type of signature. So then finally, with Eran Cohen-Barak, who did a 2-year stint in my laboratory and is now back in Israel, setting up his own lab, we actually looked at patient material, and this was also with Ellie, and we did RNA-seq of SAM patients. And we found that they also had a striking IL-17, IL-23 skew signature. And you can see here when we compare with keratinocytes that have been treated with these cytokines, that these signatures pop up, and less so the Th2 type cytokines. So Eran decided to treat some of his patients with ustekinumab, the IL-23 inhibitor that's used commonly in psoriasis patients, and could see that after 12 weeks and even more after 10 months, that they had a striking positive response to this, to ustekinumab. And in addition, if we looked before and after, we could see that there was prominent IL-23 staining in the epidermis of these patients, which was then reduced dramatically after treatment with ustekinumab. So I think you can see now that in my career, with the help of my collaborators and the team that I've built, we've gone from molecules to cells to tissues and finally to patients. And if I had gone to a cell biology department, I venture to guess that I never would have had this fantastic opportunity. And we've also shown that desmosomal diversity allows for new functions in complex vertebrate tissues, including differentiation, stratification, the formation of the barrier, and paracrine signaling. Finally, I'd like to give a shout out to some of the other people in my lab who I've not yet talked about, but we're continuing to look at the role of inherited desmosome in disease and in skin development and in Darier's disease. We have a project on Darier's disease, and Sophia and Cody are the leaders in that effort, and in melanoma, which I didn't have time to talk to you about, but it's very exciting. So finally, I think we're almost at the end of the meal. And so for dessert, I would like to thank all of the members of the Green Lab, present and past, and all of our collaborators in our cardiac work, which I didn't talk to you about, our skin work, and all of our funding sources. And I'd like to send out a message that we really need to stay connected, and it's never more important than right now. And so I'm so glad that we've had this opportunity to connect with you all over the world. And I'm hoping that in the very near future that we'll be able to start visiting in person again, as we have done so joyously in the past. And I'm looking forward to seeing you all very soon. So with that, I'm at the end of my talk, and I guess I turn it back over to Ellie and those of you who want to manage the questions.
So thank you very much, Kathy, for this exciting presentation and impressive career. I think my video should start. Yes. And I want to encourage everybody to provide us with questions for the discussion. And One question that arose to me was, is desmoglein 1 also regulated by inflammation, that it's not only regulating signaling of keratinocyte and also inducing inflammation, but what happens if microbial stimuli induce inflammation, and how does it affect desmoglein signaling and expression? And could this be also a link how certain trigger factors may push bullous pemphigoid, and what are your thoughts about this?
That's a really fascinating question, and it's one that we've been talking about a lot in the lab, and recently had a visit from Yasmin Belkade and talked to her about that. I think it's known that, and I think Rich Gallo has shown some of this, that there are bacterial products that actually elicit or at least are associated with loss of desmoglein 1. And in fact, we've treated cells, keratinocytes, with peptidoglycan and see that desmoglein 1 goes away within an hour or so. It's just gone. So it's possible that its loss is triggered by microbial products, and that might then kick in some of what we view as protective responses. And so we think that the UV or bacterial products or other types of stresses might cause this temporary loss of desmoglein to induce this inflammatory or other kinds of signatures, which are at least in part protective. That's our idea anyway. But then chronic loss could have a sustained kind of inflammation that might lead to things like you see in SAM syndrome, for instance.
Of course, it's pemphigus and pemphigus diseases that are important. And the question, so far I do not see any other questions that arise, but yeah.
I mean, there is a question about pemphigus. I might add that we actually did RNA-seq analysis also on, I think it was 7 pemphigus foliaceous patients, the lesional skin epidermis. And interestingly, they do not have this really prominent IL-17 signature. They have a slight one, but it's not prominent. Their signature, they have changes in expression of genes involved in keratinization, and actually a lot of changes in genes involved in actin cytoskeletal function, but they don't have the same kind of inflammatory signature that we see in SAM syndrome or in the mouse. So it appears to be that the autoimmune antibody responses are a little bit different. It's a slight signature, but it's not prominent, which is interesting.
Yeah. So clinically, this threshold function of cell adhesion, so there may be some degree of adhesion present, and then trigger factors may drive increased loss. And this could be, on the one hand, microbial challenge. It could be UV challenge. It could be inflammatory challenge. Do you also see pharmacological triggers that regulate
that's
molecules, desmogleins, hemidesmosomes?
Well, certainly they're very sensitive to various kinds of external growth factors, for instance, can cause the internalization and loss and downregulation of cadherins in general, but certainly of desmogleins as well. And I'm sure that there are probably a number of pharmacological drugs and things that would cause those kinds of changes. There are certain kinds of cytokines that are produced under stressful conditions, or in cancer, for instance, that will cause the turnover of desmoglein 1. And we're really interested, actually, we can show that desmoglein 1 is lost around pigmented lesions early on in even before you see frank melanoma. And we can show that that melanoma cells make secreted factors that cause desmoglein 1 to go away. And we think that there's crosstalk then between the desmoglein deficient keratinocytes and those melanoma cells. So yeah, I think the answer to your question in general is yes, but we haven't done a systemic study of what kinds of drugs might cause changes in expression of desmoglein 1.
So we finally got questions. My one question is, why does psoriasis not blister if it's similar to desmoglein knockout?
Well, I think actually, so we did some staining of psoriasis patient tissue, and you do definitely see a loss of desmoglein 1, but it's patchy. But you see a loss of that, and you see things that go along with it, like loss of connexin 43, which we've shown there's a connection there. But there are also changes in other adhesion molecules, of course, in psoriasis and also in the mouse. But in psoriasis, you're going to have a lot of desmoglein 3, 2, et cetera. And actually in the mouse, we see an upregulation of desmoglein 3. It goes, instead of just being restricted to the basal layers, it goes way up into the superficial layers, but it's not enough to counter the dramatic loss in the superficial layer of desmoglein 1. But in psoriasis, it's not a complete loss. So I wouldn't expect to see frank blistering in psoriasis, but there could still be some changes in signaling, and maybe desmoglein loss is contributing to that. That would be interesting.
Yeah, one
so
question.
Katerina, I'm sorry.
One question from Australia. Would you predict that forced expression of desmoglein 1 would reduce UV-induced epidermal proliferation and inflammation?
Well, it's asking a lot from it, but it would be an interesting experiment to try. We actually did in vitro, we made 3D cultures, treated them with UV, different doses of UV, and we could see the responses, upregulation of MAP kinase signaling and other responses due to UV. And then we actually did put desmoglein 1 back into those cultures to see whether it would increase the kinetics of recovery of those UV treated cultures, and it did. But that was in vitro with the 3D reconstructed cultures. So whether that would work in an in vivo setting with sunlight, it would be an interesting thing to think about.
I think that we are a little bit.
Okay, the final thoughts.
Yes, well, unfortunately, there are actually now many questions, so we will actually forward you all those questions and maybe and then send answers to the chat later on. I just would like, Katie, to thank you again for this amazing lecture. I mean, I think that there are many people who are actually congratulating you on the chat and on the Q&A function. Not only really science at its best, it also shows how relevant basic research can be to medical practice. And I think this is something extremely important and relevant to all of us. So just before closing, I'd like just to remind everybody not to forget to connect for the next episodes of the ESDR Kitchen. December 9, we have a freshly baked session with 3 outstanding speakers, Philip Starkel, Stefan Askamp, and Francesca Capone on novel papers dealing with psoriasis and staph aureus infections. January 13, we have a relatively hot debate at our next sweet and sour session on the association between atopic dermatitis and cardiovascular complications, Sarah Brown and Sinead Langan. And with this, I'd like to thank you all very much for having been with us today. Take care and bye for now. Bye-bye.
Thanks, everyone. Bye-bye.